Welcome to the Deep Dive, where we take complex source material and really pull out the essential high-yield knowledge you need.
And today we are tackling a big one. It's one of the most elegant, complicated, and just utterly critical systems in all of biology.
The human immune system.
Exactly. This is such a foundational subject. I mean, whether you're a student getting ready for boards, a young professional who needs a refresher, or, just someone who's curious about how you stay alive.
You need a clear roadmap. And that's really our mission today, isn't it? To build a thorough but still accessible guide to immunology.
It is. And for that, we're leaning on a fantastic source. It's Basic Immunology, Functions and Disorders of the Immune System by Abbas and Lichtman.
It's really the gold standard for this. It's designed to distill all these dense biomedical facts into clear, understandable principles. And that's what we're going to do, make those principles stick.
Okay, so let's start with the big picture. The text introduces the two fundamental cooperative pillars of how you defend yourself.
Right. First, you have the innate immunity. And you should think of this as your immediate firewall.
It's always present, it's ready for action, and it responds immediately.
But it's general. It recognizes these broad microbial structures, kind of like seeing an enemy uniform but not a specific soldier's face.
And that's the trade-off, you see. It doesn't improve with repeat exposure. It's the same response every time.
Then you have the second pillar, the adaptive immunity.
And this is your precision-guided missile system. It has to be stimulated by a very specific antigen, which means, it takes a few days to really get going.
Well, when it does, it's incredibly specific. And crucially, it generates immunological memory.
Which means the second time it sees that same antigen, the response is just massive and fast.
Now, here's where it gets really interesting for me. The two systems aren't separate, are they? They're completely locked together.
Absolutely. That innate system, that first line of defense, is what provides the necessary second signals, we'll get into those, that are absolutely required to fire up the adaptive response.
So the innate response is kind of like a gatekeeper.
That's a great way to put it. It makes sure that the adaptive system, which is immensely powerful and potentially self-destructive, only gets turned on when there's a genuine microbial threat, not just, some random dust particle.
Okay, let's unpack that sentinel system then. Innate immunity doesn't even start with cells, does it? It starts with physical barriers.
Right. Your skin, the linings of your gut, and your respiratory tracts, these epithelia are your first massive shield.
And they're armed.
They're chemically armed. The epithelial cells themselves produce these little peptide antibiotics that can kill bacteria on contact. And nestled within those barriers, you've got these early warning sensors, the intraepithelial lymphocytes.
Which are often a specific type of T cell, right?
Often gamma delta T cells, yeah. They act as immediate sentinels, recognizing very general structures shared by lots of different microbes.
So when a breach happens, the cellular recognition system takes over. And this is where that concept of pattern recognition really shines.
And the stars of that show are the toll-like receptors, or TLRs.
So what makes them so important?
Well, TLRs are crucial because the innate system doesn't need to recognize every single possible bug out there. It just needs to recognize common molecular structures, things that are shared by big groups of pathogens.
These are the PNMPs, right? Pathogen-associated molecular patterns.
Exactly. So you can think of a TLR as a kind of security key that only fits the locks on these generic microbial surfaces.
Can you give us a quick example, just to make that concrete?
Certainly. Take TLR4. It is specifically designed to recognize bacterial LPS or endotoxin, which is a major part of the outer wall of gram-negative bacteria.
Okay.
Then you have others like TLR3, -7, Medix of 8. They're usually inside the cell in these little bubbles called endosomes, where they're perfectly positioned to spot viral nucleic acids like double-stranded RNA, which you really only find when a virus is replicating.
So when a TLR locks onto its target, what happens? It triggers an alarm.
It triggers a huge alarm sequence. The signaling pathways inside the cell activate these powerful transcription factors, NF-kappa B and IRF3.
And that leads to what?
That immediately leads to the expression of inflammatory cytokines, things like TNF and IL-1, and the really rapid production of type I interferons, which are just essential for putting all the surrounding cells into an antiviral state.
This alarm brings in the first wave of cellular responders.
The phagocytes.
And first on the scene are the neutrophils.
The rapid deployment force, that's right. Neutrophils are your most abundant circulating white blood cell, normally about 4 to 10,000 per microliter of blood.
But that number can shoot up.
It can shoot way up. The moment that alarm goes off, their numbers can jump to 20,000. They are the absolute first cell type recruited to a site of infection. They're fast, but they're also short-lived.
Then you have the monocytes and macrophages.
Right. Monocytes are what circulate in the blood, but once they move into tissues, they change. They differentiate into these tissue-resident macrophages, like Kupffer cells in the liver, and they become these long-lived, powerful cleanup crews.
Now, you mentioned the process of getting these cells from the blood to the infection site is a key thing for learners to grasp. The migration cascade.
It is. It's a beautiful 4-step choreography, and you need to know it.
Okay, let's walk through that choreography. Step 1.
Step 1. A resident macrophage in the tissue senses a microbe and immediately starts dumping those inflammatory cytokines, TNF and IL-1, into the area.
Okay, so the alarm is sounded locally.
Step 2. These cytokines activate the endothelial cells, the cells lining the nearby blood vessels, and cause them to suddenly express these sticky molecules called selectins.
And what do the selectins do?
Well, that's step 3. The leukocytes whizzing by in the blood start to weakly bind to the selectins, and because of the blood flow, this weak binding causes them to roll along the vessel wall. It slows them way, way down.
And finally, they stop and commit.
That's step 4. Chemokines, which are also being released, bind to receptors on those rolling leukocytes. This instantly activates a second set of adhesion molecules on the leukocyte called integrins.
And these are stronger.
Much stronger. They flip into this high-affinity, super sticky state that mediates firm adhesion, the cell stops, and then it squeezes right through the endothelium into the infected tissue. That whole process is what causes the redness and heat we all recognize as inflammation.
Wow. Okay, so finally, in this innate lineup, we have the natural killer, or NK, cells. Their function is fascinating because it seems to overlap a bit with the adaptive system.
It really does. Their main job is to kill infected and stressed host cells, which is a lot like what the cytotoxic T lymphocytes do. And they also secrete IFN-gamma, which is a major macrophage-activating cytokine.
But the aha moment the text points out is how they get activated. It's not about spotting an intruder, it's a balancing act.
It's a total balancing act. They have activating receptors, which spot molecules on stressed cells, and they have inhibitory receptors.
And the inhibitory receptors recognize our own cells.
They recognize MHC class I molecules. Think of MHC class I as a license plate that says, I belong here, I'm a healthy cell. If the NK cell sees a valid license plate, the inhibitory signal wins, and the NK cell moves on.
Wait, so if a cell is infected with a virus and it's trying to hide, what does it do to its license plate?
It hides it. This is a brilliant piece of coevolution. Viruses often try to suppress MHC class I expression to hide from the very specific CD8 plus T cells.
What are you doing so?
They remove the brakes from the NK cells. The NK cell sees a missing cell, a cell that should have that license plate but doesn't, and it immediately kills it. It's a perfect example of this constant host microbe arms race.
That sets the stage perfectly for adaptive immunity. So segment 2. T cells and cell-mediated response. Their mission is singular, dealing with intracellular microbes.
And this is a critical distinction. We're talking about bugs that survive inside phagosomes, like mycobacteria, or even worse, ones that escape into the cytoplasm itself, like viruses or listeria.
And to find those hidden threats, T cells need help from the antigen presentation system, the MHC molecules.
Right. And you have to know the two major platforms, MHC class I and class II.
Okay, let's clarify. MHC class 1.
MHC class I is the platform that displays peptides from proteins made right there inside the cell's cytosol. A bit of the cell's own protein gets chopped up by the proteasome, shipped into the ER by a transporter called TIP, and loaded onto a class I molecule.
And as recognized by?
Exclusively by CD8 plus T cells. These are the cells that become the killers, the cytotoxic T lymphocytes, or CTLs.
So class I is basically a report on the cell's internal health. What about MHC class 2?
Class 2 is for sampling the outside world. It displays peptides from things the cell has eaten extracellular proteins it's ingested via endocytosis. This complex is then recognized by CD4 plus T cells.
A helper T cell.
The helper T cells. So to put it simply, CD4 plus helpers see outside threats, CD8 plus killers see inside threats.
And to get a naive T cell going, we need those two signals we mentioned at the start. Signal 1 is specificity.
Right. Signal 1 is the T cell receptor, the TCR, and its co-receptor physically recognizing that peptide MHC complex. But if that's all that happens, the T cell actually gets shut down. It becomes anergic.
So where does the immune system safety check come in?
That's signal 2, co-stimulation. The antigen-presenting cells only express molecules called B7 when they've been activated by those innate signals we talked about. The B7 molecules have to bind to a receptor called CD28 on the T cell.
No B7, no activation.
No B7, no activation. The T cell assumes the antigen is just a harmless self-protein, and the response is shut down before it begins.
But once both signals are received, the T cell goes from standby to fully active. And that requires a very rapid internal signaling cascade.
It's a rush of biochemistry. That initial recognition leads to phosphorylation of these things called ITAMs, which recruits and triggers the key enzyme, ZAP70.
What's the most important outcome of that cascade for us to remember?
I'd say focus on the pathways that lead to transcription. The NFA pathway is critical for making cytokines. The cascade activates an enzyme called calcineurin, which allows NF8 to go into the nucleus and turn on the genes for IL-2 and its receptor.
And there's a practical link there, right?
It's a huge one. The immunosuppressive drug cyclosporine works by inhibiting calcineurin, which stops T-cell activation cold. It's a cornerstone of transplant medicine.
And all this activation leads to specialization. The CD4 plus helper T cells become the conductors of the entire immune orchestra.
They really do. They differentiate into different subsets. You get Th1 cells, which make IFN gamma. They're all about fighting intracellular pathogens by promoting what we call classical macrophage activation, turning macrophages into superkillers.
Then you have the Th2 cells.
Right. They make IL-4, IL-5, and IL-13. They focus on barrier immunity, fighting off large parasites like worms. They do this by driving IgE production and activating eosinophils in mast cells.
And they also handle cleanup.
They do. They promote alternative macrophage activation, which is less about killing and more about tissue repair and remodeling, so they manage the recovery phase too.
And the final primary subset, the Th17 cells.
They secrete IL-17, and their main job is to shout for reinforcements. They recruit neutrophils and monocytes to the site of infection, which makes them very effective against certain extracellular bacteria and fungi.
Okay, that brings us to segment 3, humoral immunity. This is the world of B cells and antibodies.
And their mission is entirely focused on extracellular microbes and their toxins, things floating around outside of cells.
And the key difference is what they can see. Unlike T cells, which need those little peptide fragments, B cells can recognize everything.
Proteins, lipids, carbohydrates, and they see them in their native three-dimensional shape. It's a much broader recognition system.
The weapon they produce is the antibody, that classic Y-shaped molecule.
Exactly. 2 heavy chains, 2 light chains. The 2 arms of the Y are the Fab regions, and that's where the specificity is. The very tips are hypervariable, especially a region called CDR3.
And the base of the Y is the FC region.
The FC region is the business end. It mediates all the effector functions, like binding to receptors on other immune cells or activating the complement system.
So when a B cell sees a protein antigen, it needs permission to launch a full-scale attack. It's a T-dependent response.
Right. The B cell eats the antigen, processes it, and presents little peptides on its MHC class II molecules. It's like the B cell goes to an activated helper T cell and says, "Hey, look what I caught. Is this dangerous?"
And the helper T cell gives the go-ahead.
It does. It gives 2 critical signals. First, physical contact through CD40 ligand on the T cell binding to CD40 on the B cell. And second, it secretes specific cytokines.
And those signals drive the B cell to not just multiply, but to get better.
To optimize the antibodies. The first optimization is heavy chain isotype switching.
So that's changing the function without changing the target.
Precisely. The B cell changes the FC part, the constant region, to get a different function. It might switch from IgM, which is good for a first response, to IgG for systemic protection. Or, if the T cell provides IL-4, it'll switch to IgE for parasites and allergies. The specificity stays the same.
And the second optimization is even more powerful, affinity maturation.
This is just incredible. It's like the immune system runs a high-speed evolutionary competition. The B cell's antibody genes undergo somatic mutations, creating slight variations.
And only the best ones survive.
Only the ones that bind the antigen more tightly get the survival signals. This all happens in the germinal centers of lymph nodes. You end up with these incredibly high-affinity, super sticky antibodies.
So what do these optimized antibodies actually do?
3 main things. First, neutralization. The antibody just physically binds to a virus or a toxin and blocks it from interacting with your cells. This is the main mechanism that makes most vaccines so effective.
Second is opsonization.
The eat me signal. Right. Antibodies, especially IgG, coat a microbe. Phagocytes have SC receptors that see that antibody coating, and it triggers them to gobble up and destroy the bug.
And third, they can activate the complement system.
Via the classical pathway. When IgM or IgG binds to a pathogen, it kicks off this cascade of proteins that leads to more opsonization and ultimately the formation of the membrane attack complex, or MA, which punches holes directly in the microbe.
So our final segment covers control, failure, and evasion. Let's start with self-tolerance. How do we prevent all this power from being turned against ourselves?
It's maintained at two levels. First is central tolerance, which happens while the lymphocytes are developing. In the thymus or bone marrow, any immature cell that reacts strongly to a self-antigen is usually deleted through apoptosis. It's called negative selection.
But some must escape.
Some always escape, so you need peripheral tolerance. Out in the tissues, if a mature T cell recognizes a self-antigen but doesn't get that mandatory costimulation signal too, it becomes anergic. It's functionally shut down.
There's also another mechanism, right? Deletion?
Yes, through something called activation-induced cell death. If T cells are activated over and over again, they can be triggered to undergo apoptosis via the fast death receptor pathway.
And then there's the specialized police force,
Hmm.
the regulatory T cells, or TREGs.
TREGs are so important. They're CD4 plus THAW, they have high levels of CD25, and they are defined by a key transcription factor called FOXP3. Their whole job is to actively suppress other self-reactive T cells.
And if you don't have them?
If you have a mutation in the FOXP3 gene, you get severe systemic autoimmunity. It really underscores how critical they are for keeping the system in check.
When tolerance fails, or the response is just misdirected, we get hypersensitivity reactions. The text outlines 4 types.
Type I is your immediate allergy. Think hay fever. It's driven by IgE and mast cell degranulation.
Type 2 is antibody-mediated, so IgG or IgM targeting your own cells.
Right, like in Graves' disease, where an antibody stimulates the thyroid hormone receptor.
Type 3 is immune complex disease.
This is where antibody-antigen complexes get stuck in blood vessels and cause inflammation. The classic example is the kidney damage you see in lupus.
And finally, type IV is T-cell mediated.
So this is when the T cells themselves are the ones causing the damage, either directly or indirectly. Think contact dermatitis from poison ivy or chronic graft rejection.
This brings us full circle back to the evolutionary arms race with immune evasion. Microbes fight back.
They absolutely do. They have evolved specific strategies to defeat every defense we've covered. Take listeria. It has a protein that lets it break out of the phagosome and replicate freely in the cytoplasm, completely hiding from the macrophage's killing machinery.
And we touched on viral evasion with MHC earlier.
We did. Viruses like herpes simplex block the TAQ transporters, so viral peptides never even get loaded onto MHC class 1.
And cytomegalovirus, or CMV.
It goes a step further and just removes the class I molecules from the ER entirely. It's a direct countermeasure. But this is where the system's brilliance comes in. While the CD8 plus T cells are blinded, the NK cells see that missing self signal and step in to kill the infected cell. It's just a constant back and forth.
The complexity of immunology really is about appreciating this specialized deployment, from the instant response of the innate cells to the memory-driven, highly tailored response of the adaptive system.
It's a remarkable integration. Before we wrap up, let's pose a little reinforcement exercise based on these critical distinctions we've talked about.
Okay, let's make it a clinical challenge.
All right. Imagine a novel bacterium is discovered, and this bacterium can escape the phagolysosome and replicate freely in the cytoplasm of a macrophage. The question is, which type of T cell response, CD4 plus or CD8 plus Li, is ultimately required to eliminate the reservoir of this infection, and by which MHC pathway would its antigens be presented?
Okay, the key here is the location. The microbe is in the cytoplasm. It's become an intracellular threat, hidden from antibodies. So the macrophage's own killing machinery can't get to it. That means you need to kill the whole host cell to eliminate the reservoir. So the answer has to be CD8 plus cytotoxic T lymphocytes, the CTLs.
And the pathway.
And because the bacterial proteins are being made in the cytosol, they'll be processed just like the cell's own proteins. They'll be presented on the MHC class I pathway.
Exactly. Understanding that intracellular versus extracellular distinction is just. It's the core principle of cell-mediated immunity. And I think the ultimate takeaway here is that immunology is never a finished story. Every single successful defense we have is met by a microbial countermeasure.
Phagolysosome escape, MHC downregulation.
Right, and this constant, elegant evolutionary struggle is precisely why the immune system is so robust, so layered, and so endlessly fascinating to study.